Hello, everybody. Welcome back to another episode of the Lil Dudes Insect Academy podcast. I'm your host, Bradon Coy. Wait, before we get started, I just wanted to say thank you for tuning into this episode of the Lil Dudes Insect Academy podcast and just ask you to please hit the like button below or click the subscribe button if you're watching on YouTube, or follow this podcast if you are listening. That really helps the show out. But also, if you want to help us further, then join our Patreon. We have an awesome community that is growing there. Or if you want to make a one-time donation, we are a nonprofit foundation, so head to our website. Both the Patreon and the website will be linked down below. But without further ado, back to the episode. And I'm super excited to be here with Dr. Véronique Martel, and we're going to talk about awesome research that she has done in Canada and really all over the world. She's worked on parasitoids, and she is currently a research scientist for the Canadian Forest Service. And we're going to talk about all this awesome stuff she's worked on and what she does right now. So welcome to the show, Véronique. Thank you so much for joining me. Go ahead and introduce yourself to the people. Thanks so much.
Yeah, well, thanks for having me. So, I'm a research scientist with the Canadian Forest Service, as you said. So, I'm based in Quebec City, in Canada, at the center called the Laurentian Forestry Center. So, I do research on different kind of insects. It's usually around insects that are causing problems for the forest. In Canada, and so trying to understand them better, sometimes trying to anticipate where they will be and what kind of damage they can do, and eventually try to manage them to reduce the damage they could do.
That's really cool. And I really like to highlight different careers in entomology on the podcast. And so I'm super excited to have you on because forestry is a really cool thing that we haven't talked about a whole lot on the show before. So it's really exciting. I mean, let's just jump right into it. What does this position that you're in right now kind of entail? Like, walk us through some of the systems you're working on right now.
Yeah, so for forestry, well, I work for the Canadian government, so of course one of the goal is to try to mitigate the impact that insect would have on forestry, which is of course a major economy for the country. But we also have an ecological aspect of it, trying to maintain the forest as healthy as possible. So of course we do have some insects that are native, so that have always been in Canada or in North America, and they might have outbreak cycles. So every 30 years, for example, they will have real impact, huge impact in the forest, but also regenerating them. One of the issue that's really important is also the exotic species that we get. So of course, with the international trade, we get more and more insects coming from other countries. And
Right.
sometimes in these countries, they don't have any major impact. But once they get in our forest with the trees that don't really know them, so cannot defend themselves, and our natural enemies who don't recognize them as potential prey of hosts, then these exotic species might do quite a lot of damage, and that's a concern that is increasing a lot.
Yeah, and one of the systems that you mentioned previously that you have worked on that kind of comes to my mind with this is the emerald ash borer. So tell us a little bit about, like, I think that's a really great example of like how devastating and invasive species can become. So tell us a little bit about that system and what you've learned about them.
Yeah, so that's usually the example we use when we talk about exotic species. And one of the reasons is that it attacks a tree, of course, so it has an impact in forest, but it's having a lot of impacts also in urban forestry. So most people in North America know the emerald ash borer because there's a lot of ash trees that have been cut in cities. So we really see the impact more for that species compared to other forest species. So basically, the emerald ash borer, it's a really tiny insect that come from Asia, where it's not really an issue. But when it arrived, it was found in 2002 at the same time in Canada and in the United States, so at the border there. So normally it would attack only stressed trees, so trees that have for any reason add
Hmm.
another insect species, some stress, drought,
Hmm.
whatever. So it would need a tree to be stressed. But what happens here is that they're actually able to attack healthy tree. So they could come to any ash tree, and the female would lay their eggs, and then the tiny larvae coming out of the egg would go underneath the bark, and then when they feed there, the problem is that they block the sap from moving, in the tree.
Right.
And because of that, then the branches will start dying because they don't get the nutrients they need. And it happens quite quickly. And because it's hidden under the bark, we often realize it's there after maybe 3, 4, 5 years. So the insect
Mm,
is already established and the damage is done when we do realize that it's there. So that means that we cannot eradicate it. We have to live with it.
mm.
It's just spreading more and more, but it's here and it's gonna stay here because we have no way to eradicate it.
And just want to make sure that I have that correct. So the reason that they're such a problem here is because, and the reason that they're able to attack healthy trees is because the trees that are native here don't have a natural built-in defense against them. Is that right?
Exactly, because they haven't co-evolved. So it's like a new threat for them. Just like if we do have a new disease, we don't know, our system is unable to fight because it doesn't recognize it at first.
Mm.
So yeah, the lack of defense of the tree is one aspect, and the other one is the lack of natural enemies. So we have a lot of other insects or the other animals that would feed on insect, could feed on
Right.
emerald ash borer or
Mm-hmm.
parasitized emerald ash borer. That would be the case in their native range. But here, these insects or animals, they don't recognize emerald ash borer as a potential prey or host. So because of that, they survive more in our tree and they don't get killed. So that's the perfect mix for an exotic to survive. And that's the case with most exotic species that become invasive.
So, okay, so what other things are you working on right now, or is it like general, like someone reports a issue in the forest and you go and explore it and you have like experience with like all the bugs, or like, or do you work on, or are you researching something specific?
Yeah, I usually have a quite specific research project, but of course we're sometimes being contacted about new issues.
Mm-hmm.
So that's often how a new research project might start. So for example, currently I am working on emerald ash borer and on the biological control. So we're using these natural enemies I mentioned. We're using these ones from its native range that we've bring here. Of course, we make sure that they don't have impacts on our native species.
Mm-hmm.
That's really,
Mm-hmm.
really important. So that's a long process to do that and get it approved. And then we bring them here and have them attack emerald ash borer here, hoping that we won't be able to eradicate it, as I said, but hoping that we can at least reduce the population and maybe somehow find a new balance, hoping that the ash trees will maintain in our forest. Probably not in our cities. I don't think any cities will plant ash tree anymore, but that in the forest they will maintain there. So
Mm-hmm.
that's an example of something I'm currently working on. Yeah.
Yeah, so we've talked about this quite a bit on the show before, but you know, this classical biological control is where you introduce natural enemies to help combat this issue, right? And I'm pretty interested in like the process of that. Like in Canada, you've got this, you know, you've got emerald ash borer, and so what you're doing is you go back and you're doing parasitoids, right? Okay, yeah. So you go back to their native range, find parasitoid or multiple that attack them and lay their eggs, you know, in the larvae, I think, and you know, introducing them to this new range. I'm kind of interested, like you talked about, it's a long process. I'm kind of interested in like what the process for that is, because you are now introducing another species and you need to be certain that it's not gonna, you know, have a similar effect to the, you know, the thing you're trying to fix, right? You need to make sure that it doesn't disrupt the native, this ecosystem that you're introducing them to. So I'm interested in like, what is the process of getting this approved by the Canadian government so that you can release this new species? That's really
Yeah.
interesting to me.
Yeah, you're absolutely right. It's really important. And just to clarify, in the case of the Emerald Ash Board, this project was started by a research team from the States. So they're the
Okay.
one who went and researched in the native range and identified species that could be suitable. And then the researchers in Canada joined the project, the American project, about it because of course insects have no borders. So if something is introduced in the States, it will eventually come. But each country has their process, which is kind of similar, but it has to be done in both countries. Well, each country has to do it for its own regulation and to be allowed to introduce. So Among the things that are need to be done, a lot of experiments on what we call non-target impacts. So that means that we take these parasitoids and we need to put them with different of our native species, insect species, and see can they attack our species that are in our ecosystems, because we don't want them to attack our species. That's really important. And parasitoids, depending on the species, a lot of parasites can be really specialized. So they will attack only either one species or one, genus or family. So a small group of insect species. So that's something we're looking for when we do classical biological control. So we want parasitoids or predators, but we want enemies that will focus as specifically as possible on a single species. So
Right.
we would do tests in the lab, so put them in presence of our species, of a mix of different species to see which of these species will they attack preferably. And these are the kind of studies that need to be done and then presented to the regulation organization that will then look at that, and they could say, well, it's not good enough. I want more proof, more studies. They could say, well, no, it's not specialized enough, so we don't want it. Or they could say, yes, I'm convinced you have enough scientific proof to proceed with it, and that's how it's done. So it takes quite some time, but I think it is important to do it carefully
Mm-hmm.
because we don't want to make things worse. That's for sure.
Right. And once you've released it into the wild, you can't undo that. Right.
Exactly.
It's out there forever.
Yeah.
Mm-hmm.
Yeah. Absolutely.
Yeah, this is really really interesting. So what like step in the process are you guys with that right now? Like, has it been approved? Is it a wasp or
Yeah, there are 4 different species of wasps that have been approved in both countries, Canada and the United States. But in Canada, we're only releasing 3 of them. The reason is that one of those species is actually not a big fan of our winter in Canada. So it's a little bit too cold for one of these species. So because of that, we're not releasing that one. So we have 3 that we're currently releasing. Um, 2 are attacking the larvae, and 1 is attacking the eggs. So we're targeting different life stages, hoping that we at different stages we can bring more mortality, so reduce the population of emerald ash borer at different points in their development. And now we've released them in several provinces, so we're kind of following the spread of EAB and trying to follow that with the parasitoids. But once it's introduced and established, then they do their job. So we don't have to keep releasing it. The idea is become established, and then they will come out, look for new EAB, and they will even disperse with EAB when they expand their range. So that's the hope that eventually we'll be able to just let the parasites do their job and hopefully save a few ash trees.
so what's the like releasing process? Like, do they breed them in captivity and then release them, or do they collect them from Asia and bring them over, or
So at the beginning, they collect them from Asia, bring them and have them as a rearing in the lab.
okay.
And now we're using the North American colonies to release in the different sites. So because we also need to make sure we know which species we have. So we never take something from the wild and release it. It has to go into a colony to make sure it's pure and we didn't mix it up with another species because we don't want, again, to introduce something we're not sure of. So yeah, so that's how it's done. It's rare in the lab. It means that to rear the parasites, of course, we need to rear the emerald ash borer.
Mm.
So
Hmm.
it's not the easiest rearing, I'd say, because it develops in trees.
Yeah.
And the adult of emerald ash borer needs foliage, needs leaves to feed. So these are
Mm.
not the easiest ones to rear, but they are rearing in the States, and we have one for 2 of these species in Canada.
Wow, that's really cool. Yeah, really, really, really interesting. So, how long have you been doing this? How long have you had this position for?
I started in 2011.
Okay, wow, yeah.
Yeah.
Um, that's really cool. And what have you been like working on mostly Emerald Ash Borer like the whole time, or have you kind of switched around a little bit?
Now, when I started, emerald ash borer was not in Quebec City. And because of that, I wasn't working on it because when you work on an exotic species, if it's not where you are when you're located, you will need like the proper containment lab, and we don't have that in my center. So because of that, I
Mm.
didn't work on that when I started. I switched to that when it arrived in Quebec City. When I started, what I started working on, and I'm still working on it, is a native species, which is the spruce budworm. So it's a caterpillar. It's native, so I've always been around feeding on the buds of spruce and balsam fir. But what makes it interesting or like a well-studied species is that it has outbreak cycles. So every 30 to 40 years, you have the rise of an outbreak, and the levels of population can be really, really high. What we often show to people to give an idea of how many insects they can be. So the caterpillars will be in the buds, but then as any caterpillars, they will become, in that case, it's a moth, so it's a nocturnal species,
Mm.
and they can fly very long distance. So depending on the nights, if the weather is good for it and you have the winds for it, they can travel like hundreds of kilometers, but you would have like
Wow.
millions or billions of moths traveling to a point where I have colleagues who are looking at the weather radar images and they can see the clouds
No
of moths
way.
traveling. So it's really impressive. If you're there the next morning on the path of that migration, you could find like millions of moths like on the ground, half dead, or kind of flying a little bit around. They would be attracted to light, so we often would see that in car sailors, like used cars place where you have
Right.
a lot of lights,
Mm-hmm.
a lot of
Mm-hmm.
spotlights, so they would be attracted to that. And so it's really, really impressive. And of course, it does kill trees. It's natural, it's normal, and it's part of the cycle of the forest. Trees get older, and at a point, they become more susceptible, they will die, and that leaves place for new species and younger for the regeneration to come. But it still has an impact, of course, on the forest economy.
So, do we know what the reason is for these outbreaks? Is it like what you said, which with the age of the trees, like all the trees are now really old and they're more susceptible? Is that what we think it is, or?
It's been studied for a while, and some people would tell me, well, I mean, don't we know everything there is to know about Swiss bloodworm? And you could think that it would be the case, but a cycle being like 30 to 40 years,
Yeah.
that's one researcher's career.
Mm-hmm.
Every researcher will see only one outbreak cycle in its life, pretty much. Well, not in its life, but
Wow.
in its career. So that
Right.
makes it longer to test hypothesis and to study. So we don't know everything, and there's a lot of different hypotheses, and I won't go into details on all these hypotheses but we do know some factors that will have an impact on the outbreak cycles. So the age of the forest is one thing. So younger trees will not be as susceptible. So we usually see that in forests that are older, that it would have a lot of balsam fir. So balsam fir would be the main species that will be attacked. And I know that it's called spruce budworm. Saying that it prefers balsam fir to spruce sounds kind of weird,
Yeah.
but the timing of balsam fir is better for the species. But they probably realized when they named the species, they were probably interested in spruce as a resource in the forest. So that's probably how it got its name. But having older trees, a lot of balsam fir are some of the factors. The weather, of course, some years the climate is better or not. You have more mortality during the winter, for example. It can have an impact. And then what we realized is that migration is probably also a trigger. So you kind of always have the insects somewhere in the country because it's in from west to east in Canada. And it's also in the northern part of the United States. So you would always have it somewhere. So probably that when you have a fly, some moths coming in and laying eggs, it kind of give a boost to the population that will allow the outbreak to start. Because when you're not in an outbreak and you don't have a lot of these insects around, there are two main things that makes it low in the low state. a lot of mortality. So the caterpillars are being preyed on by birds or other insects, spiders, things like that. They have a lot of parasitoids attacking them. So a lot of the caterpillar won't survive to the moth stage. So you have there one factor that's important. And then the moths that you get that becomes adults, they are so few adults in the forest that they have a hard time finding each other, males and females. And because of that, the success of mating is low. So a lot of females will not be able to lay eggs because they didn't find a mate to fertilize their eggs.
Hmm.
So a lot of mortality and not a lot of success in reproducing makes it staying quite low. But if you have a boost in the population, let's say you have a migration coming in, a lot of females will lay eggs, so you'll have a boost, and then natural enemies might not be able to bring the population down. You have more males and females, they can find each other, mate, so it means more eggs are being laid, and then you have the outbreak starting, the population rising, and then until it reach the peak where you would, well, trees will start dying, so the resources will be a problem for them. If trees are dead, they don't find any food.
Mm-hmm.
The caterpillars
Mm-hmm.
cannot feed. They die more, and then the natural enemies will also, at one point, kind of get the control over the budworm population and bring it back down, and then you might not see it for another 30 years.
Wow. So it's like a snowballing perfect storm, basically. That's really interesting. Wow. Yeah, that's really, really cool. So I want to talk about your college career a little bit. So tell us a little bit about your experience with your various degrees and what you worked on and where you went for those.
Yeah, so I did my undergrad in biology at in Montreal. So I'm not a trained forester. I'm really a biologist by training. So I did my undergrad in biology there, and then I did my master and PhD at McGill University, but with a researcher from Agriculture and Agri-Food Canada. So in a federal lab, and that's when I started working on parasitoids. So that's when my love for parasitoids started and never stopped. So parasitoids are just fascinating.
Yeah.
I mean, they lay their eggs inside another insect, a host, and they will develop inside. But they
Mm-hmm.
have so many features that are impressive. So, for most of them, for example, the female can decide if she wants to lay a son or a daughter. So, when she does lay the eggs, she can make the decision of fertilizing or not the egg. And that's only one example, but to me, it's just fascinating that an insect could control. Can you imagine if human could do that? That would probably not be a good idea, though. But
Probably not, no.
so I did my graduate studies on parasitoid species. I was working back then on Trichogramma, which is an egg parasitoid, used a lot in agriculture, in biocontrol. So you can buy it depending on which crop you're growing. You would have a different species that would be good to control whatever pests you might have. They're also present in forests. So I did my graduate studies on more the agricultural side, but in my current job, I did use Trichogramma to against Spruce budworm. So I did some biocontrol assays using Trichogramma, but a species that would be specialized in forest and that would be native to our Canadian forest. So that was the graduate studies. As I said, fell in love with parasitoids, but I wanted to try something else. I wanted to explore a bit after that. So I first went to Sweden for 2 years, where I did a postdoc. I wasn't working that much on parasitoids there. I work on a Lepidoptera species, so a moth
Okay.
that is a problem, among other, in Egypt. So I went to Sweden to study an Egyptian insect. So original.
Okay. Yeah.
Why not? So I did really, really fun stuff. I changed a bit the kind of things I would do. I did some fun side project there where I used mosquitoes that I would put with caterpillars to see, and the mosquito can actually feed on caterpillars' hemolymph. So the hemolymph being kind of their blood.
Huh.
So that's
I've
something that's
never
not
heard of that.
well known. Yeah, and if you look in the literature, scientific literature, you would see really, really old papers mentioning that. But I had no idea it existed, but somehow we found it out and we decided to try. Funny thing is that I was making experiments in cages. I would have cotton plants, the caterpillar, and
Mm-hmm.
the mosquitoes in there. But if I
Mm-hmm.
wanted the mosquitoes to attack the caterpillars, I had to stay quite far from the cage because they were more interested in my blood
Right.
than in the caterpillars. But they did feed on them, and they were able to produce eggs, the mosquito females, after that. So
Wow.
that was a fun project.
So it was kind of more of a last resort. Like, oh, we need a blood meal. This works.
Exactly. That's what we think, of course. That is kind of a last resort if there's nothing else, or for example, if the weather is pretty rainy, so mosquitoes wouldn't fly a lot, something like that. They might, again, last resort. Probably not the best food for them, but they can succeed on caterpillars hemolymph. Yeah, that was a fun side project that I did. I mean, I published one paper on it. It wasn't the main thing in the lab, but I mean,
Yeah.
in research, we also like to have fun sometimes, and that's the
Yeah,
kind
of
of thing
course.
that you do. So that was my first postdoc. A second postdoc after that, I went to France, where I went back more on the parasitoid side, but for climate change questions. So they had different species that would spread north with climate change, and we were looking at the impact of then the competition with this new species moving in and competing with local species. All that on parasitoids of Drosophila, so the fruit flies.
Yeah, so you've done all the parasitoids, the fruit flies, emerald ash borer. Oh, that's really cool. And I heard this interesting argument that Hymenoptera, which include the parasitoids, the parasitoid wasps, are the order with the most species. I've heard that argument. Currently described, it would be the beetles, right? But I heard the argument that they think it could be Hymenoptera because you could make the argument that there is a parasitoid for almost every species of insect, right? Or there could be that we just haven't described yet. Because that's what we see is like most parasitoids are very, very specialized, right? So they're specialized towards a specific species or genus or something like that. So I just thought that was really interesting. Like, what are your thoughts on that? Like, do you think there could be way more parasitoids than we've described?
As a taxonomist,
Mm-hmm. Mm-hmm.
but yeah, there are a lot of parasitic species. We are discovering new ones. There are still in some cases where we know we've found females but no males yet. So is it because there are no males or the males just so different? There are species where males would be attacking one host species, but males and females, sorry, would develop in different host species.
Mm.
And then you have the parasitoids that we call hyperparasitoids that are attacking a parasitoid that's in a host. So I mean the level is just there's no limit to the complexity of the system.
So
And
there
I'm
are
just thinking
parasitoids of
of
parasitoids.
parasitoids exactly.
Yeah.
So just for Spruce budworm, we're talking probably about 60 to 100 species of parasitoids that can attack spruce budworm or that are hyperparasitoids of the spruce budworm. So that's for only one species. So I could imagine that makes a lot of species. And I mean, you're talking Hymenoptera, but parasitoids are some of the Hymenoptera are parasitoids, but you have other Hymenoptera that are not parasitoids. So you have the bees and the wasps and the ants, and then you also have the phytophagous ones also. So the sawfly are
Mm-hmm.
also Hymenoptera, not
Mm-hmm.
parasitoids and not social insect. So I can imagine that Hymenoptera is so diverse. And for insects in general, we keep discovering species, which is amazing. We would think that we know everything, but we just
Right.
discover more. And I think molecular biology and genomics is helping with that because sometimes we don't see the difference. But then if you go deeper and look using these
Mm-hmm.
new tools,
Mm-hmm.
might realize that they were different species that we just were confounding. Yeah, it's
Right.
fascinating.
Oh, that's so cool. Yeah, so I mean, you went through your college a little bit, a little bit of an overview, but like, tell us a little bit about first your master's research. I was interested in that. Tell us a little bit more about that. I'm interested in hearing about what you did there.
Yeah, so during my master, I was working, as I said, on Trichogramma, so the egg parasitoids. They're really, really tiny. I mean, they attack insect eggs, so that gives an idea. But we're talking maybe a little less than 1 millimeter in
Wow.
length, so that's pretty small. But that makes it easy to study because you can have a really large rearing in a lab and it doesn't take much space. So they're kind of easy to rear, which make them a good biocontrol agent, but also a good a good model species. And I was looking into the reproductive strategies of these insects. So one of the things I was looking, as I mentioned, the female can decide to lay a female or a male, so son or daughter, when they lay eggs. And one of the thing I was looking back then is to see some factors that would impact their decision. So one of them that is well known is competition. So if you have several females in the same place looking for resources for hosts, they will lay, let me make sure I don't, more males. So we call that sex ratio, the proportion of males and females would tend to get more balance, about 50-50. But if a female is alone, she will lay just enough sun to inseminate all of her daughters. So this is one of the species, the group species where brothers and sisters do mate together, and
Mm.
there's no genetic impact on that. So because of that, they are there because yeah, she doesn't want the females are the one that will attack new hosts, right? So if a female wants to bring her gene to the next generation, she needs more females because they're the one that will lay eggs in more hosts. So because of that, if she's alone, and she knows that her progeny will just mate with each other, she will lay just enough son. So it's usually
Hmm.
about 20%. So two males for eight females would be about the ratio that they would do if they're alone. But then I was testing if you put more females, how does it change your sex ratio? And then I was also testing if you put females of another species, if she could perceive that they're not competition because they cannot mate. The two different
Hmm.
species cannot mate
Yeah.
together normally. So that's the kind of thing I was looking at to try to see how she will make her decisions based on our immediate, I'd say, habitat.
It's really interesting. And um, what were some of the things you found? I mean, you mentioned that you found this sort of somewhat conscious as much of a conscious decision as a bug can make. But this uh this act of sort of trying to balance the scales in a certain way for the best success. Is that something you found, or was that like already in the literature beforehand?
It was in the literature, like as a general theory, I'd say. But yeah, I did find that they do adapt their sex ratio depending on competition. And I was working with several species. The response was different depending of the other species. So sometimes they would perceive the other species as competition, and sometimes they wouldn't, depending on which species. So that's, I mean, I don't have the full explanation for that. One of the hypotheses was depending if in natural condition they would encounter that species. If they do, it means that they probably develop some ability to detect the difference, to smell that
Mm,
they're not the same species. As of course in the lab, I mean, we can make really artificial conditions. So we might expose to species that would never encounter in the wild. So that sometimes explains some of our results, the fact that
yeah.
it's not something that was meant to happen.
Yeah, we're just out of curiosity, were those species that you introduced closely related, or were they like,
They were the same genus.
oh, same genus? Okay.
Same genus, yeah.
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 So could could you see, like, not see, but like, could there be some interbreeding, like hybridization that could
Yeah.
be a part of competition?
We sometimes you'd see attempts by males to these interspecific females, but there would never be any progeny produced. So they
Mm,
might sometimes kind of get confused. Again, we often put them in closed space, so the male sometimes has a choice between trying to court the wrong species or do nothing. So if they perceive pheromone from the female, so the smell that females will emit to attract males, they might sometimes try. But usually that's kind of how we define species. That's
right.
the limit when they cannot interbreed. Although
Hmm.
we know that there are cases when it can happen, but often then the progeny produced will be sterile. They won't be able
Yeah.
to reproduce. Yeah.
Yeah, that's really cool. Yeah, so going back to your PhD studies then. Tell us a little bit about that.
Yeah, that was
What
fun.
was that?
So I stayed on the same system. So Trichogramma again. I was in the same lab, but then I kind of shifted a bit because during my master I was looking at females strategies for reproduction. And when you look in the literature, you kind of have this kind of taken for granted that the females will make wise, if we can say from an insect, but wise decision because they don't have a lot of eggs. They need to be sure how they invest these eggs because they're precious. And you would often see that males have an unlimited supply of their gametes, which are the sperm.
Mm-hmm.
So they won't choose, they don't have to choose. They will just try to mate with as many females as possible because
Much
they're not
less
limited.
picky.
Exactly. So I during my PhD, I was looking at the male's perspective, kind of trying to show that it's not completely true. That even though, of course, males have more sperm than females have egg, they still need to invest these carefully. So I spent a lot of time dissecting males and counting sperm, and dissecting females that were mated and counting the sperm to know. So I was
Really?
really measuring how males would invest sperm in the different females that they would encounter. And they
So,
do. They do have to.
yeah.
Well, they do make some decision. They would invest more in some females than others. They will get picky also in their mates. Not as much as females maybe, but then depending on where they are in their life. Like when they're closer to the end of the life, the insects will tend to be a bit more choosy. So it was interesting. And when I was going to conferences and talking about my results, a lot of people would just tell me, yeah, but no, males aren't, they're not picky. They don't have to be. It's like, well, not as much, but they still are limited in their resources, depending on the species again. Some species will
Right.
produce sperm their whole life. Some species will have a limited number, and they will have to do with that their whole life.
Yeah, yeah. I mean, the act of, mating with one female in itself is a choice, right? I mean, it's an energy. They're expending energy to be with a certain female as opposed to another one. So there is some level of investment there, even though it's way less than the female. Yeah, but that's interesting to think about because that's kind of what I was always taught was the females are way more choosy and the males kind of just go and mate with as many whatever females as they
Yeah,
can, right?
yeah. And depending on the species, and of course I'm mainly talking about insects and parasitoids because that's my expertise. But then you would have species. So there's the concept of sperm competition, which is the
Mm-hmm.
competition of sperm within a female. So
Mm-hmm.
female of several insects and for example parasites, they have an organ called the spermatheca. So the females only have that, and that's where they store the sperm that they receive.
Mm-hmm.
And then that's how they can choose to fertilize or not the egg when they oviposit because the eggs are not already fertilized inside of her. So she will release
Right.
sperm every time she lay an egg if she wants a female. Because we remember the sons are not fertilized, so male parasites don't have a father. They only have a
Right.
mother.
Mm-hmm.
And so there's this concept that if you have sperm for more than one male in the spermatheca of the female, these sperm will compete. So, and depending on the species, it's not, they don't do it the same way. So you have species where you have last male precedence. So the last male to inseminate a female will have more chances of having its sperm fertilizing eggs. And you have species where it's the first male that has more chances. And you have even have species where a male will remove the sperm from the previous mating and replace it with its own. So this
Wow.
this whole dynamic that might change depending on the species, and that makes it the strategy will be different for the male then. So if the first male is the one that wins, a male will want to go for virgin females because they want to be first. But if
Mm-hmm.
the last male wins, then it's different. Going for a female that's already mated becomes kind of strategic because you want to be the last
Right.
one
Yeah.
because the last one will have more. So yeah, that's what I did during my PhD. So it's pretty
That's
fun.
so interesting. You mentioned really quickly, and I just wanted to echo this, but you mentioned really quickly that the parasitoids, only female parasitoids, have a mother and a father, but the males only have a mother. And I just wanted to mention that that's true for all of Hymenoptera. So all ants, bees, wasps, and sawflies have this system where the unfertilized eggs develop into males and the fertilized eggs develop into females. And then you mentioned that The female can choose when she lays an egg whether it will be fertilized, becoming a female, or unfertilized, becoming a male. And I wanted to mention that that's really important also for the social insects, right? So if there's a queen, if there's a queen ant or a queen bee or something, and she wants to make a worker, right? All workers are female. She will fertilize the egg to make a worker, right? So I just wanted to mention that that's how that works. That's a thing even in social insects where she gets to choose because males and females have very different roles in those systems, just like they do with your parasitoids. So really, really cool stuff. But yeah, I just wanted to mention that.
Absolutely.
Yeah. So jumping ahead, like, what are some like things you want to work on in the future? What are some big plans? Whether it's research or travel or big plans for the future of you.
Well, I always stay, as I said, sometimes people contact us or me about new issues that I may or may not be aware of. So I like working on different things. I mean, Spruce Budworm will always be there, so I'll probably always work on it. But I like also looking at new emerging insects or concerns. And one of the things I'm working currently, it's been a few years, but that I didn't realize was an issue, is a close relative to the emerald ash borer. But that one is called the bronze birch borer, and it's a native species here. Similar lifestyle to emerald ash borer, but because it's native, it only attacks the stress trees, only the birch.
Mm-hmm.
It's not a problem here. We would typically see them when if you have, for example, a forest where there was a drought or some stress like that. So you would have a bunch of birch trees that would be stressed, and then it will come in and attack because they're secondary. So they attack stress trees, kind of like emerald ash borer in their native range. So it's not a problem here, right? But what I realized recently is that there are some European countries that are worried about it because they don't have it and they don't have to receive it because it could become invasive there.
Same situation as the emerald ash borer.
Exactly. So it's kind of, the opposite situation. So emerald ash borer is a problem for us here because it's exotic for us. But we do have indigenous species that could become invasive in other countries. And we don't talk about these as much. We don't think about it, of course, because it's not a problem for us, but it could become. So because of that, I've started working on that and trying to understand better and also try to get the knowledge and the scientific knowledge necessary for example, the industries to make the proper phytosanitary measures. So when we do export wood, just like when we import wood, we want it to be insect free. And ways to do that could be to debark, so remove a part of the wood under the bark. It can be to make heat treatments. So classical would be 56 degrees for 30 minutes. For most species, it's enough to kill any insect developing inside. Oh, but these are all measures that can be certified and they're agreed by the different countries. So the exporting country and the importing one. So we're working on these questions currently to try to help the industries, but also CFIA, the Canadian Food Inspection Agency. So they're the one that would negotiate, let's say, these measures with other countries. And we're trying to provide the necessary scientific data for them to make the right decision to protect the European countries from getting our insect, but also trying to make the measures that are, let's say, the more restrictive you are on the measure and the highest the cost it is for us. So that's something I'm starting to work on that I really wasn't working on before, and that is quite far from what I did during my studies, but that I think it's really interesting and have an impact for the economy of the country.
Yeah, and kind of like a preventative, before the issue
Yeah,
becomes an issue, right?
because we should always try to prevent the exotic, not
Right.
fight them when they arrive. Ideally, we would prevent them from arriving. And again, if we want to prevent them from arriving, we should prevent us from sending
Mm.
other
Mm-hmm.
species. Yeah. So it's totally different. I did some pretty fun stuff for that project. One of the things we wanted to know is how deep are the larvae beneath the bark? Because if you
Okay.
want to know how much wood or bark you need to remove, you need to know exactly where the insects are.
Yeah.
And typically with these insects, when you want to find the larvae, the technique is called debarking. So we will slowly peel the bark until we find the insects or the galleries from the larvae. But when you do that, of course, you destroy your sample because you need to remove the bark to find the insect. So what we did is we used the CT scan, so the actual medical equipment that we call tomodensitometry, if we want to have the fancy word. And so we have logs that we pass in the CT scan, and it will give you it detects the density of anything. So we can see them in the tree. We can see the larvae or the galleries of the insects. And because the sample is still complete with the bark and everything, we're then
Right.
able to go and measure precisely how deep they are. So we're able to say, well, of course, the average, they're in average that deep. But when you want to prevent exporting insect, you don't want the average. You don't want to be able to remove the insects most of the time. You want to remove every single insect. So then we're able to say, okay, so the deepest insect we found was at that depth. So we need to remove that much wood under the bark if you want to make sure that insects are dead. So that was quite it was a fun technique to use.
Yeah, that's
Personally, I,
cool.
I didn't know that we could use CT scan for science, but they're actually used in different kind of areas.
It's so cool. Yeah, I've heard of like micro CT scans for even like the anatomy of insects, and it's really cool to me. And you don't really think about it like, oh,
No,
that
exactly.
that technology can be used for all kinds of things.
Yeah, but they're
Yeah,
expensive machines, of course.
of
So
course. Yeah, yeah, yeah.
we didn't buy one. We had a collaborator that had access to that, and we would rent the machine by the hour for our
Right.
samples.
Yeah, yeah, yeah. Yeah, anything else you wanted to talk about? Like stuff you want to do for the future or stuff you're excited about?
Yeah, I do a lot of community science in my research.
Okay.
So just as a reminder, that's when we work with the community, so citizens, other organizations, to help us study something. So we do that a lot in different projects. And I wanted to mention it because this help and this collaboration with communities is so precious to us. Because I mean, we had a project with Spruce Budworm where we wanted to have pheromone traps in most part of the Spruce Budworm range, and we had like up to 500 volunteers at some points in Canada, in the States,
Wow.
but we also have a French island, Saint-Pierre-et-Miquelon. So we had technically three countries because of France having traps set up. People would look at these traps. Often the kids would be the one doing it in families. And then we would have really great data because we could have like 3 times per week at the moths that were caught. People just put them in the freezer in a paper bag and then they send it to us at the end of the year. And that's data we wouldn't be able to get ourselves because just traveling by
Right.
vehicle to all these traps would cost way too much and take
Hmm.
way too much time. So community science is amazing. And we even have one of our projects where, thanks to a citizen, we discovered a new exotic species that was discovered for the first time in North America in 2020. And it was during the pandemic, so traveling was restricted.
Oh
But
yeah.
somebody posted a picture on iNaturalist of some zigzag pattern of defoliation on an elm tree, and several people saw it and realized that that was an insect called the elm zigzag sawfly. So another Hymenoptera, but Phytophagous one,
Hmm.
and it was not known for North America. So that was thanks to one person who took a picture,
Wow.
posted it on our website, and we were able then to confirm that it was a new exotic species. So people observing, taking pictures, posting stuff is precious. Really, really.
That's really
So
cool.
it's amazing when people out there are curious about what they
Yeah, for
see around
sure.
them.
For sure. Yeah, so sort of shifting away from your research and more about you, like what are some of your favorite hobbies to enjoy in your spare time, maybe aside or related to science?
Yeah, so I like I have diverse interests. I like crafting in general. So I do quilting at home. I do stained glass too.
Oh
I like
wow.
to try all the different things when I can do this kind of one day workshop on this or that or wood carving or so I like trying this. I feel like being really rational and scientific in my job makes me want to do something a bit more creative. Although research is really creative, but I
Yeah.
like to kind of express my creative side outside of work. But I also like a lot video games, board games. Big fan of horror movies. So
Oh really?
I mean, my water glass I use at work is one from a Stephen King classic movie.
Oh yeah.
Yeah, but I also love museums, theaters, plays. Oh yeah, I like cultural stuff in general.
That's so cool. What like what video game system do you have?
I have a PlayStation 5 currently. I don't game on computers on PC, which for some the real gamers will
Mm-hmm.
think it's a sin, but yeah,
Mm-hmm.
I have a Switch, Nintendo Switch and a PlayStation 5 currently.
Awesome. It's so cool. Yeah, do you ever find like your crafting moving into the bug realm? Like you quilting with bugs or like
Well, of course, I often will use a pattern or designs with insects in it or bugs or animals in
mm-hmm.
general. So, oh yeah,
Mm-hmm.
yeah. I cannot. My heart is is filled with bugs, so there's no way I could do without it. Yeah.
It's so awesome. Oh, that's really really cool. I think that's about it. Is there anything else you wanted to say before we wrap it up? I think we covered just about just about everything, but we could talk a lot longer if we want.
Maybe just a last message to teenagers and kids. Just
Hmm.
go for whatever you like. Don't get restricted by what society tells you should or shouldn't do. So I'm in forestry. I'm a woman in research and forestry, which are kind of two areas where there are often not a lot of women. But
Hmm.
I mean, I'm just doing what I love, and I think that that's important. Then people around me are supportive, and they see that I'm doing. I mean, I'm having passion in what I do, so I just encourage people to do whatever they want.
That's so cool. And yeah, and it's pretty obvious you've had, quite the career doing what you love and doing really cool and like impactful stuff, right?
Hmm.
Invasive species are becoming more and more of an issue with travel. The more trade we have, the more this is going to become an issue. Yeah, I just think what you're doing is really really cool, and I like to highlight all the different careers that young students can take, and this is another great example of that.
Yeah, and with passion, I mean, when you love what you do, it's so easy to do it. So yeah,
Absolutely. Yeah, well, Veronique, thank you so much for chatting with me. This was super cool. Nice to see you again, and thanks again for taking the time to chat with me about this.
it was a pleasure. Thanks for having me.